everyone. Welcome to today's live webinar, Score No Compromise Protein Quantification on Lunatic. I'm Megan Manzano, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by BioPharm International and sponsored by Unchained Labs. I would like to share a statement from our sponsor before we begin. Unchained Labs is all about helping biologics and gene therapy researchers break free from tools that just don't cut it, unleashing problem-tackling solutions that make a huge difference in the real science they do every day. That's their mantra, their promise, and they own it. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. I would now like to introduce our speaker for today. We are pleased to be joined today by Ross Walton. Ross Walton is a senior application scientist for biologics at Unchained Labs. He has published works on single particle protein analysis by transmission electron microscopy and the interaction of the innate immune system and viruses in cancer. He did his undergraduate work at UC Berkeley and earned his PhD in molecular genetics and microbiology from Duke University. Thank you so much, Ross, for joining us today. Please get us started.
Thanks for that introduction, and thanks to everyone who's joined me today to talk about no-compromise protein quantification on Lunatic. One of the things I've been trying to do this year is have more people over at my house so we can share dinner together. But it can be pretty challenging to do that because everyone has different food preferences and different dietary restrictions. Vegetarian, vegan, carnivore, gluten intolerances, food allergies, they're all valid and they're all important, but it can make it nearly impossible to find one menu that suits everyone. Finding the right protein quant system for every group in a biologics pipeline is kind of the same way. Everyone needs to do protein quantification, but they also have very different requirements and very different needs depending on where they are in the development cycle. For example, throughput and low volume consumption might be more important early on in development when you're testing lots of different samples and you have very small amounts of each one of those samples. Whereas later on, regulatory compliance might become more important as you go closer into manufacturing. Of course, accuracy and precision are important at every stage, no matter where you are, from the benchtop all the way up to manufacturing. Lunatic meets the needs for each department so you can unify and standardize your protein quant without compromising. That way, you can have one solution across many different departments, which makes everything from method transfer to troubleshooting a whole lot simpler. For early stages of development, it's low 2 microliter volume requirements, and measuring 96 samples in about 10 minutes is a game changer. Because the plates have a standard SBS footprint and a standard well layout, they're compatible with liquid handling robots and multi-channel pipetters, which can really let you ramp things up. Lunatic plates make all of its amazing capabilities possible. They have the same well spacing as a 96 well plate, but instead of just a well, each one is connected to a microfluidic circuit. Each microfluidic circuit has 2 fixed path length microcuvettes, which cover a wide dynamic range, from 0.03 OD to 275 OD. These circuits ensure there's no cross-contamination or evaporation. And fixed path lengths give a high level of precision and accuracy to absorbance measurements, so you know exactly what it is you're measuring. While it takes just a single step to measure something with the Lunatic, just load and go, the instrument itself actually tells a few different steps before it gets you your concentration results. After you pipette your sample into the well, it's pulled into a reservoir by capillary action. At this point, the sample can sit for up to 2 hours before reading with no evaporation. That makes life a lot easier, especially if you're running lots and lots of samples in a day. Then you load the plate into the instrument, which reads the empty microcuvettes to eliminate any absorbance contribution from the plastic, and a pump applies pressure, which moves the sample from the microfluidic reservoir into the microcuvettes, and that last absorbance measurement is taken. Here we can see exactly what sample loading looks like with the help of some fluorescein and a blacklight. In 2 wells, we've already added 2 microliters of fluorescein. As you can see, to load another sample, you simply add 2 microliters into that input well, and then the sample wicks into the plate. Here's what it looks like before and after a read happens. You can see the fluorescein in the microfluidic reservoirs on the left, and on the right, the samples have been pulled into the microcuvettes. You might notice that one of the cuvettes is a lot brighter than the other, and that's because one of them has a slightly longer path length than the other, which also means that there's more volume of the sample of the fluorescein in that microcuvette. So it's brighter and it's more easily visible. This combination of 2 different path lengths is part of what gets Lunatic its phenomenal dynamic range. The most common protein quantification methods simply can't match Lunatic's throughput. For example, using a microvolume pedestal-based spectrophotometer would take about an hour to measure 96 samples, but this involves a lot of hands-on time, and it really isn't as precise or as accurate as you'd like. Now, there are some methods that are extremely accurate and precise, but they also tend to be slow and will only measure 1 sample at a time. In those cases, 96 samples would take about 96 minutes at least. Lunatic accomplishes this same task in about 10 minutes. Using Lunatic is as easy as it gets. The GUI of the Lunatic client software makes it easy to pick the right application to measure your protein sample, and then visually lets you lay out your plate, or you can just import a plate layout from Excel. After you take the measurements, the data gets sent to Lunatic Analysis so you can view it, create reports, and then also export results to Excel if that's something that you need to do. That ease of use really reduces training time, which makes a huge difference for any department. Lunatic lets you max out your biologic concentrations and can measure high concentration samples across a huge measurement range with high reproducibility. This dilution series of an antibody in PBS really highlights this. The 8 replicate measurements at each dilution have UV-Vis spectra that overlap each other nearly perfectly across the full absorbance range from 0.03 to 275 OD at 280 nanometers. The replicates agree with each other, and there's extremely good linearity across this entire range with an R-squared value of nearly 1. Now, these results look pretty good, but you might be asking yourself, how good are they exactly? Lunatic brings in 2% accuracy and 1% precision for its absorbance measurements. I think anyone can agree that's pretty impressive, especially considering it only needs 2 microliters of sample. When you look at Lunatic's linearity, accuracy, and precision over a range of concentrations, they're equally impressive. This shows an IgG dilution series from 2.3 mg per ml to about 200 mg per ml. The close agreement between the target concentration and the average measured concentration shows off Lunatic's spot-on accuracy within 2% of the expected value. The low CV values across the dilution series show off its precision, typically below 1%. Delivering this dynamic range, linearity, accuracy, and precision is the advantage of using fixed path length microcuvettes. When we compare Lunatic's reproducibility to other microvolume spectrophotometers, for example, ones that are pedestal-based, it makes the results even more stark. Keep in mind, pedestal-based systems require cleaning after each measurement, but Lunatic wells are single-use, so you'll never have to worry about making messes. Here, we measured 4 different proteins at 4 different concentrations and graphed their %CV, or their relative standard deviation, to compare precision for these 4 different samples. Lunatic is precise and across high and low concentrations for many different protein types. Lunatic's accuracy and precision for purified proteins is impressive, but it also has some specific unmixed applications that's useful for dealing with cell lysates or protein samples that haven't completely been cleaned up yet. These kinds of samples can have a variety of impurities that will contribute to the overall UV-Vis absorbance spectrum, resulting in something that looks kind of like this black absorbance curve. A traditional UV-Vis spectrophotometer would interpret the entire A280 signal as being from protein and therefore would over-report the concentration. Unmixed algorithms analyze the sample spectrum and can deconvolute it to reveal the absorbance from just the protein, as well as the absorbance from impurities or from turbidity. It does this based off of the characteristic spectra of each one of these types of components. The overall shape of a protein's absorbance is caused mostly by its tryptophan residues. And this shape is actually really different from the shape of a nucleic acid's absorbance spectrum. That means that it's possible to separate these two signals from each other and then determine the concentration of the protein sample based off of the deconvoluted protein spectrum alone. The unmixed applications also report absorbance values for the impurities and for the turbidity. And that way you can tell if a protein sample needs additional purification. It also can help you determine what might be causing an unusual signal. If a protein sample has really high impurity absorbance and low yield, that might mean that you have to maybe increase the concentration of your protein inputs or change them in some other way. On the other hand, if it has really high yield and only modest impurities, then maybe it just needs an additional cleanup step. Here's the result of a protein sample analyzed with unmix. The black line is the absorbance spectrum before the deconvolution. Unmix identified the absorbance from the protein, which is shown in green, and the contribution from nucleic acids and other impurities, which is shown in blue. You can see that there's a peak at 260 nanometers in the blue line, which is characteristic of nucleic acids. In the results table, you can see that the absorbance at 280 nanometers due to the impurities. In most spectrophotometers, this absorbance would make it look as if the protein concentration is much higher than it really is. This sample has a significant amount of nucleic acid, so it might need further processing or repurification, depending on how it's going to be used in the next step of its workflow. The gray line is the turbidity signal, which can actually come from a lot of different sources. There's a lot of different things that can contribute to the turbidity of a protein sample. When we use unmix, the unmix applications also show the A280 value of the turbidity background in the table. This absorbance value would also interfere with most spectrophotometers' quantification. High levels of turbidity in a protein sample or of a cell lysate can come from a lot of different sources. Lipid contamination, protein aggregates, or sometimes even bead or resin carryover from the purification process itself. Getting some information on impurities at the same time as you get the protein concentration can help you determine if a sample needs further processing, if it needs additional purifications, and it can even tell you if it's worthwhile to do all that extra work. After all, if the protein concentration is too low, you might be better off just throwing it out and starting over again. This example shows a protein sample at 1 milligrams per milliliter with a significant amount of bead contamination. You can see that the A280 value of the gray turbidity signal is actually higher than the green protein signal. This is definitely a case of high turbidity, which will most likely interfere with downstream processes. So the sample would need to be repurified or perhaps just discarded. Spiking experiments comparing Lunatic to a system which doesn't do this kind of absorbance deconvolution is a nice way of showing exactly why it's important. A 1 milligram per milliliter sample of human serum albumin with a DNA spike-in is a nice model for some of the impurities that would be present in cell lysates. And it shows that without deconvolution, the reported protein concentration would be about 50% higher than it actually is. On the right, we have an example of what turbidity can do to protein concentration measurements. This is a 1 milligram per milliliter sample of IgG with 100 nanometer beads as a spike-in, which serves as a model for turbidity. The beads for that are used in protein purification, either as part of resins or as magnetic beads, are a lot larger than 100 nanometers. So they'll scatter even more light than the beads shown here and cause even worse turbidity, even if they're at a lower concentration. No matter the impurity, and regardless if you're working with totally pure or only partially purified proteins, Lunatic still gets the right answers. Unmix is a tried and tested component and tried and tested application that's been part of Lunatic ever since it was launched in 2017. We've talked about some of the features in Lunatic that mainly solve problems for early stage researchers. It's low volume, it's high throughput, and it has those unmixed applications. Now, high accuracy and precision are important no matter what stage you are. But as you get later in the development cycle, it becomes more and more important to have proper, independently verified standards, as well as protocols and documentation to verify and validate system performance. That's why we've collected results using reference standards in Lunatic and documented it so that anyone can check it. Uracil and tryptophan NIST standards are intended to be used to verify that the path length of a microvolume spectrophotometer is as expected. For us, that means that the fixed path length of the microcuvettes in the Lunatic plates is correct and reproducible. Remember, Beer's law is that the absorbance of a substance is equal to its extinction coefficient multiplied by its concentration and the path length. These standards are part of the NIST Standard Reference Material 2082, which includes ampules of a tris buffer as a blank, 1.4 millimolar tryptophan in the tris buffer, and 1.0 millimolar uracil in that tris buffer. We measured both standards in octuplicate across 5 different systems and found that all 40 measurements were within 1.65% of the certified value, while the mean and the median measurements were within 1.25% of the certified value. This is just another example of how reliable Lunatic is and how high the quality of the plates really is. You can use NIST uracil and tryptophan standards to show the fixed path lengths are correct and reproducible and that the plates are manufactured to a very high degree of quality. But I think it's also a good idea if we look at the overall performance of Lunatic with a standard that approximates your actual molecule of interest. NIST RM8671 consists of 10 milligrams per milliliter of a human IgG1 kappa monoclonal antibody called NIST-MAB in a histidine buffer at pH 6. When we measured the concentration of this reference material in octuplet with 2 microliters of sample in high Lunatic plates on 5 Lunatic instruments, all 40 of the resulting measurements were within 1.25% of the certified value. The mean, shown by the X in that box and whisker plot, and the median, shown by that horizontal line, are within 0.6% of the reference value. According to standards defined by the US Pharmacopoeia, analytical methods for determining drug concentrations should be within 2% of the known concentration, plus the manufacturer's uncertainty. The NIST-MAB manufacturer uncertainty is about 0.4% of the reference value. So that means an analytical quantification method should give a result that is within 2.4% of the reference concentration. Lunatic hit this mark with absolutely no problem. In addition to NIST standards, Lunatic also has the fundamentals, which let you use a built-in application to check the performance yourself. The fundamentals consists of 5 tryptophan standards with certified concentrations at 1, 2, 4, 6, and 8 milligrams per milliliter. All of these certified samples have color-coded labels, so it's easy to keep track of what they are. Now, these concentrations may seem a little bit low, but you have to remember the extinction coefficients of tryptophan are much higher than most proteins. The equivalent IgG concentration of these standards are 20, 40, 80, 120, and 160 milligrams per milliliter of IgG. When you run the fundamentals with 16 replicates, it makes absolutely sure that the system is performing well across the entire absorbance range from 20 to 225 OD at 280 nanometers. When we plot the results from measuring each standard, it shows off the great linearity of Lunatic over that entire dynamic range. You might not be able to tell, but the graph on the left side, that scatter plot, each one of those points that you see is actually the 16 independent A280 measurements that overlay each other almost perfectly. The slope and the R-squared value of that trend line are both also very close to 1, which means that the target concentration and the measured concentrations were extremely close. Lastly, if we look on the right, you can see that the spectra of those replicates also overlap beautifully. When we compute the average concentrations and plot some summary statistics, you can see that the average concentrations measured by Lunatic for each one of those standards was within 1% of the target concentration. That is, mean recovery was within 99%. You can also look at the relative standard deviations, or RSDs. It's the same thing as %CV, and you see that the RSDs were less than 1%, even at the highest concentration, showcasing Lunatic's high degree of precision. Like with all Lunatic applications, the fundamentals application is easy to set up, and the software walks you through exactly how you do it. All you have to do is fill in the certified concentration of each ampule, and then also fill in the manufacturer's uncertainty for each manual, which is also written right there on the label. The plate layout is color-coded, so it's simple to know how you're supposed to fill in that plate. Checking the results from the fundamentals is also simple, because Lunatic does most of the work for you. The software calculates the average concentration, determines a mean recovery by comparing the average concentration to the certified concentration, and calculates a relative standard deviation for each one of those standards with those 16 replicates. The software also calculates an R-squared value to show the linearity of all of the standards together. Since tryptophan has an absorbance peak at 280 nanometers, which is the same peak wavelength with a absorbance maximum as a protein, it's a great way of checking the system's performance at a wavelength that is relevant to high concentration proteins more regularly without relying on purified proteins or using up some of your precious sample. All of this data can be exported to Excel or be put into a PDF report, so it can be put into a LIMS or an electronic lab notebook. Lunatic's fixed path lengths are perfect for proteins. They deliver spot-on accuracy, precision, and linearity, and can easily meet the US and European pharmacopoeia requirements for UV vids. If that USP or EU pharmacopoeia requirement is of particular importance to you, because, for example, you work in a highly regulated environment, then there's an easy way to check and verify that the system is up to the task. The USP and European pharmacopoeia have specific requirements for the absorbance accuracy, precision, linearity, wavelength accuracy, stray light, and resolution for UV-Vis spectrophotometers. To verify that Lunatic meets those standards, we offer the Performance Verification Plate, which holds enough certified standard cuvettes to check and make sure that the system's performing up to those standards. We call the plate, plus the cuvettes, the PV kit for short. Using the PV kit is as easy as any other experiment on Lunatic, and you'll get a report for each one of those required tests, showing the results and a pass-fail for the pharmacopoeia criteria. The PV kit can be made part of an IQ-OQ for Lunatic, and Lunatic also has 21 CFR Part 11 compliant software tools available, which makes it ready for all your downstream needs. These software tools has a full data integrity check and full audit trail, as well as electronic signatures. If you're interested in automation, Lunatic has you covered. Lunatics are compatible with liquid handlers and plate handling robots, and has full robot integration options based either on Sila or using its fully functional API. Lunatic plates are also trackable with barcodes if that becomes something that you're interested in. Lunatic gets used at every stage and every scale of bioprocessing, from the benchtop to manufacturing. It has hundreds of publications in academic research and in industry, and Lunatic is proven technology that can help unify and standardize protein quantification. It's easy to use and exceeds all regulatory requirements for therapeutic protein quantification. Of course, if you're working in upstream or more research-oriented work, Lunatic has a few tricks up its sleeve with its unmixed applications. When you switch to Lunatic, you'll be filled with bright ideas about how you can meet everyone's needs, no matter if you're in the lab or in the kitchen. Thanks for your attention today, and if you have any questions, please ask, and I'll be happy to answer them either today during the Q&A session or via email.
All right, thank you so much, Ross, for such a great presentation today. We're going to go into our Q&A session. So I would like to remind our audience how to submit questions. You can do so by typing them below under the video player in the available box. So we will start here, Ross, with what is the concentration range of Lunatic?
So the concentration range actually is from 0.03 to 275 OD. Now that might not be super useful because that is the concentration range of the actual absorbance measurements. I know a lot of people tend to be more curious about what's the concentration range for my specific protein. That's something that is a little bit more difficult to say because every protein absorbs slightly different amounts of light. So and you use Beer's law in order to determine the concentration based off of that light absorbance. Since we know the extinction coefficients, the E1% for a few different proteins, we can say, like, for example, for a typical antibody, the concentration range is about 0.02 to 200 mg per mil. But then for BSA, it's 0.04 up to 400 mg per mil. So the exact dynamic range is going to depend on each protein, but the absorbance range is 0.03 OD to 275 OD at 280 nanometers.
Great, thank you so much. We have another question here that says, what's an E1%?
Yeah, so E1% is just another kind of extinction coefficient. A lot of people are probably more familiar with the molar extinction coefficient, which is epsilon. So in this, for an E1%, that's just the absorbance of a 1% solution of a protein or 1 gram of the protein in 100 milliliters of solution. It's also pretty easy to convert between a molar extinction coefficient and the E1%, just based on the molecular weight of the protein.
Great, thank you so much. We have another question here about unmix, which says, what contaminants can unmix detect?
Each unmix application is designed to detect slightly different impurities based on how common an impurity is in a given sample type. So, for example, it wouldn't make much sense to, say, call RNA an impurity when you're measuring an RNA sample. That's the simplest application, or simplest use case. So for the protein unmix application, which is called protein lysates, there it actually looks at for a few different substances, including nucleic acids, sodium azide, as well as protease inhibitors. You can actually see a full list of the substances that each one of the applications looks for on the website or in an app note that's called a look under the hood, a look under the hood of Lunatic.
Great, thank you. And we have another question here about Lunatic, which asks, can it quantify DNA and RNA?
Yes, absolutely. Yeah, super. My rule of thumb for how what you can quantify on Lunatic is basically if it absorbs between 230 and 750 nanometers, you can use, and it's an aqueous sample, you can definitely use Lunatic to measure the concentration. And even if it isn't aqueous, even if it's in some other solvent, then it's a distinct possibility you'll be able to. So in the case of nucleic acids like DNA and RNA, Lunatic uses 260 nanometers to quantify them, and then also reports some really useful metrics like the 260-280 ratio and then 260-230 ratios, which can give you a lot of information about how pure the nucleic acid is. Lunatic comes pre-installed with all of the applications that you would need in order to measure either DNA or RNA, as well as a bunch of unmixed applications that are specifically designed for nucleic acids, either DNA or RNA. And there's a ton of webinars on doing exactly that. In fact, I actually presented a webinar with Select Science a couple of days ago about nucleic acid quantification, which should be either up on the website or on the YouTube page if somebody's curious about that.
Great, thank you. And we got another question in from our audience here that says, can I use Lunatic in a regulated environment?
Yes, absolutely. So Lunatic has a 21 CFR Part 11 compliant software package that has tools for auditing as well as the necessary access controls and user access controls. You can also, of course, we talked about the PV kit, which you can use to verify that the Lunatic is performing in a way that complies with both the USP and European Pharmacopoeia criteria for the suitability of a UV-Vis instrument as an analytical tool. So yes, and in fact, we have quite a few customers who are using Lunatic in a regulated environment.
Great, thank you. We got another question here that asked about maintenance. So what kind of maintenance do you need for Lunatic?
Pretty minimal, actually. So really, there's almost no maintenance. I mean, that's the nice thing about using those plates, that 96-volt plate we talked about, is that they're completely disposable. So you use a single well, measure that one, and that's basically it. There are ways of checking to make sure that the system is performing up to standard, up to its best possible performance. And it comes with a thing that looks kind of like a 96-volt plate that can be used to make sure that the system's performing well. And it has several internal checks in order to make sure that that's happening. I think we typically recommend that you run those every 6 months to 1 year. And that's really it. There's very little maintenance, just running that plate. But that's pretty much it. There's really not much else to it.
All right, fantastic. We have another question here, which says, how does Lunatic quantify proteins in solution by mass/volume if there is a mixture of different proteins with different extinction coefficients? A big question for you, Ross.
Yeah, so what I would say is that what I have done in that situation is used an average extinction coefficient, average E1%, that reasonably accurately reflects a bunch of different proteins. So my rule of thumb is it's a concentration factor of about 10, an E1% of about 10 in order to do that. Obviously, when you have a mix of different proteins, you really can't use a UV-Vis spectrophotometer to accurately quantify every one of those proteins individually, because all of those absorbance spectra are overlapping. So you can do it by just using an approximate extinction coefficient that pretty well models all of them. It won't be exactly right, but it'll be reasonably close. At that point, it's more akin to doing something like a BSA or a colorimetric protein assay with a standard that isn't quite appropriate. So if you look at a lot of BSA assays, if you use the wrong standard for creating a standard curve, you can be between 20 and 50% off from an accurate quantification, because proteins don't necessarily all interact with those colorimetric reagents the same way. So for those situations when I do have, say, trying to measure a protein concentration in a cell lysate where there might be multiple contaminants, I'm really looking for a reasonably good estimate. I'm usually comparing it to samples from the same experiment to make sure that I'm loading the same amount of protein into a gel, for example. So yeah, at that point, you're using kind of an average extinction coefficient.
All right, fantastic. With that, we will wrap our event for today. I want to thank the audience for attending and for participating in today's event. I would also like to thank Ross so much for his time today, and our sponsor, Unchained Labs, for making today's webcast possible. We would like to ask everyone in the audience to participate in a brief survey. This survey will appear on your screen after today's presentation has ended. You'll receive an email alerting you when this webcast will be available for replay, and we invite you to forward that announcement to any colleagues who may have missed today's event. Thank you everyone for joining, and we'll see you next time. Have a great rest of your day.